pigments are there. They are generally used for the giving the color to the food component. But also with this coloring compound, they also possesses some nutraceutical functions. So these are also called the functional ingredients. So basically, bioactive are the physiological active components that possesses the health benefits beyond their nutritional worth. There, some of the bioactive components are protein, vitamins, minerals, lipids, antioxidants, phytochemicals, and probiotic bacteria. These bioactives are very sensitive to the light and oxygen and temperature, etc. This micrometers are known as the microparticles. Uh, Next slide, please. If you classified this uh, particles, there are two basic uh, next slide.
Sir, we are not getting your voice, sir.
I think there is some technical
Right now,
disturbance.
yeah, yeah. Right now, we can hear you. Yes, sir. Sir, are you talking? We are not getting your voice. बारीक आवाज ऐकू येतो पण.
आवाज येतोय आता?
Yeah, right. Now it is audible, sir. Yes, sir.
Okay.
Yes, sir.
Again, some disturbance was there. Can you move for the to the next slide, please?
Yes, sir.
There are generally microparticles micro capsules has the two components: the core material and the wall material. This sorry. Uh, Depending on that, the structure of the core material, it has been divided into the three major groups. The monocore type means it has the single core material in the capsule. Then polycore type, it has the multiple core material in the capsule, and matrix type, it has the matrix activity in the microcapsule. Next, please. This is what there are having the certain next. Uh,
Sir, sir, are you there? You are not audible. Sanjay. Huh.
Now we will move on the advantages of this encapsulation to increase the bioavailability, to alter the drug release. This is basically this microencapsulation is carried out in the pharmaceutical industry, and they everywhere in the pharmaceutical, like your capsules are there. Then some of the drugs are encapsulated and used for the disease recovery. So their drug delivery is the major, means drug release is the major component where the particular place this drug should be released there in the patient's body. So it has been a very good advantage to give the microencapsulated drug. Then we will move to the next slide, please. There are on the fundamentals. These are the basic microencapsulation process. Means there are three components: there's core material, coating material, and vehicles. This generally core materials are the in the form of the solid and liquid forms. Then coating materials are which is covering this core material, like polymers are there, waxes, resins, proteins, then um, polysaccharides are there. Then vehicles are generally aqueous vehicles are there, or non-aqueous like ethers are there. Then your petroleum, then alcohol will be used as the non-aqueous vehicle. Next, please. Now we will see the some of the phytochemicals which is used uh, which is present in the our food components, like your omega-3 fatty acids. The major sources of these omega-3 fatty acids are your fishes, like sardine is there, tuna, salmon, markel, etc. Then your fruit means the other sources are flaxseed is there. Flaxseed has the very good amount of the omega-3 and omega-6 fatty acids. Soybean is there. Oyster, it is again the shellfish. Then walnut. Then sunflower seeds are there. Chia seeds are there. These are fatty acids are generally essential for the body. This manages generally this role of these fatty acids, omega-3 fatty acids, are to manage the cholesterol in the and blood pressure, support mental health, and reduce the weight and decreases the liver fat. Next, please. Next bioactive component is pigments. These pigments generally there are four. As you know, there are three major sources are there: fruits, vegetable, and microorganisms. And there are four. It is divided into the four categories: carotenoids, anthocyanins, chlorophyll, and betaines. This possesses the antioxidant activity, prevent cancer, and possesses the antimicrobial activity. Next, please. Then protein hydroset and peptides. Uh, like peptides are generally present in the fermented foods, and hydrosides are present in the milk, fish, meat. then collagen, egg, peas, and soybean, etc. This also possesses the antioxidant activity, prevent cancer, and antimicrobial activity. The bioactive peptides are nothing but the this is present present in the fermented foods, and it is currently there are more than 1,500 bioactive peptides are reported, or it is discovered by the various scientists, food scientists, and other scientists, and this used as the drugs for to cure the some of the diseases. Next, these are the general functions of the bioactive peptides: antioxidant, immunoreactive, then increase the mineral absorption, anti-cancer, multifunctional, anti-inflammatory, lipid lowering, and there are antimicrobial also. Next, please. Then phenols and flavors. These are generally occur in these fruits. There are various sources like fruits, vegetable, tea, coffee, wine, spices, etc. Are there. There are four phenolic compounds majorly appear in the foods: the phenols, then flavonoids, then lignins, and statins. This again the role is same as that antioxidant and lower the risk of the diabetes. Then it keeps the phenols are generally used for the longer means active for the longer times. Then cocophenols are lowered for the it lowers the heart diseases. Next, please. As you know, the flavors are present in the various food components. That amyl acetate is there, citral are present in the citrus fruits, then amyl acetate is present in the baked foods. Benzyl hydrate is present in the almonds, cinnamons. Then these are also lower down the antioxidant activity. This possesses the antioxidant activity, lower down the means obesity, then anti-inflammatory and protein protecting. These are the some of the functions of the flavors. Then other bioactive components are vitamins, that is fat soluble vitamins are there, water soluble vitamins are there. Then some of the other bioactives are enzymes and microorganisms. Next, uh, these all bioactive has the sensitive against the temperature and light and oxygen. And to cover this microencapsulation process is carried out, and to, wall for the wall material, there there should be some ideal character of the wall material which is used as a wall material. That ideal characters such as the film forming ability should be there. Then peelability should be there. Odorless, it should be odorless, tasteless, and non-hydroscopic in nature, and it should soluble in the aqueous solution or ability to exhibit the phase transition. Now we will see the next some of the encapsulant material or wall material. There are divided into the three major groups, that is carbohydrate, protein, and lipid. Like cellulose are there, cellulose derivative, chitin, chitosan, starch, agar, then alginate, carrageenan, then gum, pectins. This can be used for the microencapsulation. In next few slides, we will see the where it is used. Then proteins, then gelatin, carrageenan, then wheat gluten, soya protein, whey proteins are also used as the wall material. Then waxes paraffin, then uh, acetylglyceride and salicylic resins are also used for the wall material. There are various microencapsulation techniques are used for the encapsulation of this food material. but we will see the few of them. I have enlisted the all the techniques that spray drying is there, lyophilization or freeze drying, fluidized bed drying, extrusion, then co-conversion process, pan coating, solvent evaporation is there, polymerization is there, single and double emulsion techniques is there, supercritical fluid anti-solvent method that is generally called as the SAS method, then nozzle vibration method. These are the some of the techniques which is used for the encapsulation of the products. Now we will see the one by one. This uh, this is spray drying is the most frequently or oldest method which is used for the encapsulation of the product uh, or food components or food bioactives. It is used since last means, few decades, like it is used from 1930s to encapsulate the flavors and by using the gum acacia as a coating material. Uh, it has the good advantage that it has a very low cost in the and it is material. This is used. I mean, use is very easy, and we can in very small laboratory also we can use this spray drying technique. Next slide, please. This is the ideal drawing for the spray drying, where your emulsion of your bioactive components and wall material has been pumped uh, to the nozzle, and through nozzle it is spread to the drying chamber, where this in drying chamber the heated air is passed, and the micro droplets which is formed by this nozzle has been dried, and this is collected into the cyclone, which is again there is for the collection aspirator is attached to the cyclone, so we will get the dried microencapsulate there. Next slide, please. And same process means with the spray drying, we can use spray chilling also. The instead of the hot air, if you use the chilled air, there you will get the same means, you will get the encapsulated product, but still you have to use the solvent which is easily evaporated in the in low temperature. Generally, the your enzymes, flavors, and vitamins are encapsulated by the spray chilling method. Next, please. This is the schematic diagram or diagram for the freeze drying. Uh, Generally, I have given the example for the omega-3 fatty acid from the flaxseed. This is your it is extracted. Your omega-3 fatty acid is extracted through the solvent extraction, and it is purified. And this is mixed with the gum and HCl to get the homogenized mixture, and then it is freeze dried. Freeze dried is nothing but the sublimation principle is used in the freeze drying under the vacuum, and there we will get the dried microencapsulate. Then fluidized bed dryer is there. Same process of that your spray drying has been used, but instead of this uh, your part component flowing through the nozzle, it has been sprayed, and the your air movement is in the circulation movement. In the spray drying, it is only in one direction, whereas in the fluidized bed dryer, it is in the circulation movement. So you will get the dried product, and it is collected there itself. So the air your air velocity is lower than the your spray drying method. Next, then your extrusion technology is there. Extrusion technology where the cell or core wall material is flowing in the one direction, whereas in uh, core material has been placed inside the wall material. And after the extrusion, you will get the droplets or coated material, which is having the more means uh, larger size than your spray drying product. Next, please. This is also used. This method is frequently used for the encapsulation for the various drugs, uh, and uh, also it is used for the flavor, vitamin, and color encapsulation. Next, and complex coaction. This is generally used for the encapsulation of the your bioactive, which is generally used for next previous two, which is used for the microbial purpose or the generally used for the microorganisms, where your product has been mixed with the gelatin, and emulsion is formed with the oil. And again, if you want to use the another wall material, that is gum arabic, that both material has been placed with the emulsion, and some cooling has been carried out. So you will get the suspension of the microencapsulate uh, and your microencapsules, that has been washed to get the microencapsules. Then coating, pan coating. This is generally followed for the bigger size of the molecules. Like if you, it's general example is your coated chocolates are there, or tablets are there, which is coated with the maltodextrin. Uh, Product has been placed in the pan, and the coating material has been spread to the uh, pan to get the uniform coating. That I have already told you that the bigger size particles are there to get this. Then next is the solvent evaporation. The emulsion of the wall material and core material has been uh, tried to get the uh, your microencapsules. Next, please. This is double emulsion technique. Sorry, polymerization is there. Polymerization state. Next, please. This is the again the single emulsion technique, where the only one emulsifier agent is used, and polymer. This both are used and mixed together, and the wall material means core material has been inserted into center. And then after the washing, you will get the microencapsules. This is the next task method or the supercritical method. Then solvent in then your wall material, that is polymers, are mixed, and this is dissolved to get the uniform mixture, and then this CO2 is introduced into the at low temperature, as well as at low atmospheric pressure. Then after some time, the CO2 will be get evaporated at low 31 degree centigrade, and you will get the dried microencapsulate. Some of the advantage and disadvantages of this technique: spray drying. you will get the particle size about 10 to 400 micrometers, micromillimeters. Then it is relatively very simple process. At it is very low cost in as you compare to the other method. It is very low cost. Then both hydrophilic and hydrobaric polymers can be used in the spray drying. It is considered uh, whereas if you consider its disadvantage, it's during drying chamber. If the particles are exposed to the high temperature, then charring may be occur there, and you will get the ununiform encapsulated material. Spray cooling and spray chilling, your particle size will be 20 to 200 micrometers. Then it is less, again, it is very less expensive than the other methods. But the microencapsulation is not proper and uniform in this spray chilling. Then fluidized bed drain, as we have discussed, the some size of the particles is bigger than the spray drying. You will get the 10 to 200 micrometers in the size. Here you will get the uniform particles, means uniform microencapsules. But the control of the air pressure and slight difficult in this method. Next, please. Then fluidized bed drying, freeze drying or lyophilization. This is a little bit expensive than the your spray drying, but little bit means it is 30 to 50 times more expensive than your spray drying. But the product quality and oxidation maintained and your shape of your microencapsules are very uniform. Then co-conversion, it is in the 10 to 800 micrometers in size. It does not include the aqueous and continuous phase. This makes the encapsulation water soluble component. No requirement of the surfactants, yielding the solvent free product and moderate process conditions. Then mass production is difficult by this method. Then supercritical fluid technology. Its particle size is 10 to 400 micrometers. No requirement of the surfactants, yielding the solvent-free product. Means in other method there may be a chances of the getting some traces of the solvent, but here we are using the CO2 as a solvent. So this product which is obtained by this supercritical fluid technology is free from the solvent, and the process does not include the toxic means whatever the toxic effect of the solvent will be there, that can be free from in this process, that can be avoided in the microencapsulation. All these solutes should be soluble in the supercritical fluid. Means your core material should be soluble in the product, your CO2. Then we will see the some of the wall material used for the microencapsulation of the omega-3 fatty acids. Uh, then spray drying is the process which is used for the encapsulation of the fish oil, and the wall materials are used generally used are gelatin, maltodextrin. We can use then casein, lactose, sodium caseinate, then dextrose, then salic dextrin, methyl cellulose, hydroxypolyol. Then this can be used. Then pectin, gum arabic, corn syrup, then egg white. It can be used for the encapsulation of the fish oil. Then spray drying of the flaxseed oil. Uh, then whey protein isolate can be used. Then walnut for as a wall material. Then gum arabic, lecithin, maltodextrin, whey protein concentrate, gum arabic, and the modified starches, tapioca starch, wax, maize starch can be used as a wall material. Then freeze drying of the fish oil, then sodium caseinate, carbohydrate, then egg white, gum arabic, lactose, and maltodextrins can be used as uh, for the freeze drying of the your fish oil. Then freeze drying of the flaxseed oil is generally carried out with the gelatin. Then another method is simple co-carbonization. The hydroxymethyl cellulose can be used as a wall material. Then complex co-carbonization. This arabic, gelatin gum arabic combination can be used for the uh, wall as a wall material. Then next electrostatic bilayer. This method is used for the fish oil encapsulation, and lecithin is used for the as a wall material. Double emulsion and subsequent enzyme gelatinization method, which is used for the fish oil encapsulation. Soybean, whey protein isolate, then sodium caseinate can be used as a wall material. Ultrasonic atomization and freeze drying. This titosin is used for the encapsulation of the fish oil. Then spraying of the fish oil. In the electrospraying of the fish oil, the gel corn protein can be used as a wall material. Spray granulation and fluidized bed film coating for the fish. Uh, soybean uh, soybean protein, sorry, soybean soluble polysaccharides and maltodextrin can be used as a wall material. Next, uh, there are some methods and wall material used for the encapsulation of the polyphenols, such as for the polyphenol extracted from the black carrot, then olive leaf extract, hibiscus leaf extract, soybean extract, uh, then grapeseed extract. Uh, these polyols extracted from these can be microencapsulated by the spray drying, and the wall material can be used as maltodextrin, gum arabic, chitosan, then citrus fruit fibers, then methyl dextrin starch, modified starch can be used as a wall material. Then next is the black currant extract. This polyols extracted from this black currant extract can be encapsulated with co-conversion method, and calcium arsenate, chitosan, and gelatin can be used as the wall material. Next, co-crystallization method is used for the orange peel oil, and the sucrose syrup can be used as a wall material. For the anthocyanin, uh, hibiscus means anthocyanin and orange oil, the freeze drying method can be used for the encapsulation, and maltodextrin can be used. Maltodextrin, there are various great maltodextrins are available, and that can be used as a wall material, and pollulan also used as a wall material. Then molecular encapsulation is used for the encapsulation and the furic acid, curcumin, citric acid, cinnamon leaf extract, citrus oil, and this can be encapsulated with the modified starches. Then extrusion process can be used for the encapsulation of the citrus oil, clove oil, cinnamon oil, various spices oils, and for this the corn syrup solids can be used as the encapsulation material. Uh, Sodium alginate are also used as a encapsulating method. Then electrostatic extrusion is also used for the encapsulation of the vanillin, ethyl vanillin, and the calcium alginate is used as a wall material. Next, uh, these are some of the methods and wall material used for the encapsulation of the vitamins and minerals. This For the encapsulation of the vitamin C and vitamin A, the spray drying is used by using the chitosan as a wall material, maltodextrin as a wall material, or gum arabic as a wall material. Then ferrous sulfate, some of the vitamins and minerals can be encapsulated by the spray cooling methods, and the waxes, fatty acids, water soluble polymers, water soluble monomers, and soy lecithins can be used as a wall material. Then vitamin C and iron is encapsulated with the entrapment method by using the egg polymer phospho cholesterol tocopherol can be used as a wall material. Then vitamin C can be also used. Vitamin C microencapsulation can be carried out with the extrusion and fluidized bed coating method. The maltodextrin is used as a wall material. Then vitamin A is encapsulated by using the gum acacia by co-conversion method. Then vitamin A is encapsulated with the molecular infection method by the maltodextrin by using the maltodextrin as a wall material. Next, then encapsulation of the enzymes. Proteolytic enzymes can be encapsulated by using the alginate as a wall material by liposome method. Then proteases can be encapsulated by using the chitosan, then calcium chloride. This is the combination of the chitosan and calcium chloride, and this sodium alginate and starch with the complex co-conversion methods. Then beta galactosidase lipid can be encapsulated by the spray dye by using the chitosan modified chitosan as a wall material. Alginate, calcium alginate, gum arabic can be used can be used as a wall material. Then lipolytic enzymes also encapsulated by the liposomal entrapment method by by using the alginate and carrageenan as a wall material. Next, please. Uh, These are some of the hydrocytes and peptides which is encapsulated by using the spray drying method by whereas this as a wall material the soya protein isolate, then gelatin, whey protein concentrate, alginate, maltodextrin, gum arabic, uh, then CMC can be used as a wall material. Then casein hydrocytes can be encapsulated with co-conversion method by the soya protein isolate and pectin. Then fish hydrocytes, the C beam collagen, then casein hydrocytes can be microencapsulated by the liposomal entrapment method by using the and liposomal entrapment treatment. Uh, then there is some of the wall material which can be used for the uh, microorganisms. Next, please. encapsulation of the microorganisms or their in capsules. Then this is lactic acid bacteria or probiotic strains can be encapsulated by using the alginate and its combinations. Then probiotic bacteria can be encapsulated with high amylose cornstarch. Then it can be also encapsulated with xanthan along with gallon. Then lactic acid bacteria such as Staphylococcus there are some strains of the lactobacillus. Then this can be used using the this can be encapsulated using the carrageenan and its mixture. Lactobacillus lactis can be encapsulated with the gelatin or gelatin uh, and its gums. Then probiotic mixture can be also mixtures of the probiotics can be also encapsulated by the by using the chitosan. Next, uh, now we will see the encapsulation of the particular lycopene, which is carried out in our lab. Lycopene, generally there are various sources of the lycopene. Tomato is the, of course, tomato is the major. Then guava, then watermelon and pink grapes are there. This is generally considered as the precursor of the vitamin A, and this during the processing, tomato processing, the waste is means peel and seeds are goes generally uh, wasted, and this peel consists of the most of the lycopene present in the tomato, which is thrown out, or it can be dumped as a waste material, or it can be used as a manure. But it has the very good amount of the lycopene, and this is the almost your 80 to 90% of lycopene is present in the peel itself. So, for the extraction of this lycopene, there are this we have used the pectinase and cellulose enzymes for the extraction, and for the purification, we use the crystallization method for the purification of this lycopene. This purified lycopene is encapsulated by using the gum, gelatin, and sucrose. These are the some of the combination levels we have used for the encapsulation of the lycopene. Uh, for the core to wall ratio, we have used one part of the core material and two part of the wall material. The wall material is the combination of the gelatin and sucrose in the proportion of the 1:1. We have used three combinations: 1:1, 1:4, and 1:6. In spray drying, this is followed. This is carried out in the spray drying, and we have fixed the inlet temperature. It is 160, 170, and 180 degrees centigrade. Then sucrose. Sucrose to gelatin ratio also we have changed. That is 6 is to 4, 7 is to 3, and 8 is to 4. Then if you uh, this encapsulation efficiency, if you consider the encapsulation efficiency of sucrose and gelatin, then we will find that the uh, optimum is 4 to wall ratio when the 1 is to 4, then inlet temperature is 180 degrees centigrade, and sugar to gelatin ratio is 7 is to 3, then you will get the more encapsulation efficiency and encapsulation yield. Next, please. This is the graphs for the encapsulation yield. Then next, this is the SCM These are the SEM images of the encapsulated and unencapsulated, means without encapsulation lycopene. First, A is showing the lycopene without encapsulation, and another one is the next one is the lycopene encapsulated with the sucrose and gelatin combination. First, when you see the without encapsulation, you will find this structure like sawdust or some lumps will be there. And whereas in encapsulation, you will get the uniform structure of the encapsulated material. Next, as compared to the when we see the lycopene retention in the storage, uh, during storage, the first graph shows the lycopene retention in uh, unencapsulated, means without encapsulation lycopene. It is dropping down after the 21 days, means their retention will be very low after the 21 days, and you will find the means in we have used the five different conditions for the storage. This is we have used the open condition, means samples are stored in the open condition, then airtight condition, sunlight in dark condition, room temperature, and refrigerated conditions. So, in all these conditions, uh, the lycopene sample without encapsulation shows the very low retention. Uh, Means its stability is around 19 to 21 days, whereas the encapsulated material shows the very good retention after 42 days also. Means it is showing the about 90% of retention in the encapsulated material. Same conditions were used for the encapsulated material. For this, we have used the sterilized glass bottle with the different color and different conditions. Next, this was the some conclusion of the study. The optimized process conditions that pour to all material 1 is to 4, then inlet temperature 180 degrees centigrade, and sucrose to gelatin ratio was 7 is to 3 for the encapsulation of the lycopene by the spray drying method. The stability of among the encapsulated material, the refrigerator condition shows the more stable lycopene than the other material. And this could be helped to improve the storing stability of the lycopene. Next, then encapsulation anthocyanin. Uh, Next, uh, this next please. We have used as kokum as a source uh, for the extraction of this anthocyanin. Kokum is generally grown in the Sahyadri Hills, and which is also having the high nutritional value. That fields are used for the preparation of the kokum butter. Then your uh, pulp is used for the preparation of the juices. Then syrups. Then skin is also used for the preparation of the amsuls, which is used for the as a acidulas for the different traditional dishes. And this is also a rich source of the anthocyanin, which possesses the antioxidant and antimicrobial activity also. Uh, uh, in some cases, yes, in some cases, during the processing, this is seeds are goes waste, which can be uh, recovered, and which is used for the extraction of the butter, which is also has the high nutritional value, and which is also used in the cosmetic industries. It is used for the skin improver. Uh, this is we are more interested in the waste which is generated during the processing for this fruit. This waste is generally consists of the 60 to 70% of the your kokum seeds, and other material is about 30 to 20 to 30%. Before. This is anthocyanin is extracted with the acidified water, and which is again purified, and this anthocyanin extract is mixed with the maltodextrin, and this uh, maltodextrin to means anthocyanin ratio is about 1 is to 3. Then spray drying is used. This during the spray drying, 140 degree centigrade is used, and the feed rate was 2 ml per minute. Then you will get the spray dried anthocyanin. These are the another applications which can this microencapsulation techniques can be used in the uh, your medicine, in veterinary, household, personal care, then biotechnology, pharmaceutical industries, then chemical industries, agriculture, waste management, textile, then photography. This is used, food, trade material, medicine. This can be the other applications of the encapsulation technique. This can be used to protect the bioactive component, which is having highly sensitive to the oxygen and sunlight and uh, temperature. This is a small summary of presentation. Thank you. Any questions?
Dear participant, does anyone have questions? Just write it in chat box.
Due to some technical interference, we are not able to give the proper presentation. So sorry for that.
May I ask?
ते अनशेअर करा की.
Yes. Ethan, do you have any questions there?
Just wait, sir.
Okay, okay.
One question is from Sudhir, sir. Sir, any
Uh,
technology transfer available for encapsulation?
for the encapsulation of this anthocyanin and your omega-3 fatty acids, lycopene, we can give the technology transfer for them.
Okay, sir.
My number is shared in the first slide. We can give it to him so we can talk about.
Yes, sir.
Yes, sir.
I think there is.
Participants, I think you are more hungry, I think so. Is there any question in your mind? You can post it in the chat box. Else,
Think there is nothing is there. Technology. So there is only one there.
yeah, I'm putting this Google form for your attendance, which is most important and frequent question for everyone. Okay.
Okay.
So, Dr. Ranveer sir, I'm very much thankful to you for accepting our invitation and giving such a nice lecture, such a knowledgeable lecture to all the participants. which will be helpful to you. Have the so many papers in this field, so it's your knowledge will be definitely useful and for them as well as to if somebody want to do the research that one, he may contact to you based on this one. So now, if in no question is there, now I request Sri Sheetal sir to give the vote of thanks.
Okay. So thank you very much, sir, for very informative session on microencapsulation. You already explained types of capsulation, its advantages, and its different applications in different industries like food industry, pharmaceutical industries. You explained fundamentals of microencapsulations like coating materials, different types of coating vehicles. You explained different types of bioactive components and its functions, and then you explain microencapsulation, different microencapsulation techniques like spray drying, freeze drying, extrusion technology. So the session is very informative, sir. So thank you once again for this session.
Thank you for giving me the opportunity for the
Okay, sir.
Thank you, sir. Participant, let us meet by 3 p.m. for the next lecture. Thank you. Thank you, sir.